Hey there, this is Biology Lesson 3.02, Fluid Mosaic Model. That's an odd sounding word. Well, where does that come from and how does that apply to the picture that's on the page? Hopefully, this looks familiar. This is what we talked about in 3.01. So we have our phospholipid bilayer. And we talked about why that forms, and we're looking at basically a section of a cell membrane or a plasma membrane. We said in the last video, it means the same thing. Why do we have this fluid mosaic model? Model just means that's what it is going to look like. It's a model of the membrane. But fluid mosaic, a mosaic is a type of picture where you take little bits. So maybe you did this in elementary art class where you took little pieces of construction paper, put those together, and formed a larger picture. Fluid means it can move or it's mobile. And so what you have here is it looks like lots of little pieces were put together, and these pieces can move. This is not a fixed membrane. And think about it, phospholipids are mostly fat. Is fat really rigid? No. That gives us a structure that's flexible, but has some stability to it. And we have some structures there that help make it stable, some additional things. So that's where the fluid mosaic model comes from. So we're going to talk about, I think, 5 or 6 different structures here. Let's start off with the structures that help the membrane stay together. So let's talk about these by category, by function. So some of these structures help the membrane, M-E-M-B-R, help the membrane stay together. And I'll color code these. So the first one we're going to talk about, I'm going to color code it in pink. And you have a black and white picture, so this will be helpful. We have these little tiny molecules here. And there's a little diagram up here. I don't know if you can read it or not, and it says cholesterol. So these guys are cholesterol. Which, by the way, cholesterol is a fat. And all fats are nonpolar. So that kind of makes sense, right? If you're going to be if you're going to be embedded with a bunch of nonpolar, remember the fatty acid chains are nonpolar, you better be nonpolar too. But what this does is it helps if the membrane gets a little bit warmer or a little bit cooler, it doesn't come apart. Without this cholesterol, the membrane kind of just falls apart if it gets warm or cold. So I know they always talk about having cholesterol drugs to keep your cholesterol down. That's a specific kind of cholesterol. You actually need cholesterol because if you don't have it, your cell membranes won't hold together. So let's add the note. Helps the membrane with temperature. So if we have temperature swings, it will stay together. I'm going to try to focus that a little bit. I think that helped. The other structure that assists the membrane are these, and for me they're blue, so I'll kind of do them in blue. These are called microfilaments. And this is our second organelle. So microfilaments are an organelle. They're an organelle. They're protein strands. So they're long, thin proteins. And by the way, this is inside the cell, and this is outside the cell. You'll notice they're only found on the inside of the cell. Well, the reason is they help support the membrane from the inside of the cell. So they act kind of like tent poles. You ever put up a tent, you know that the canvas is really flexible, but the poles are strong and stable. So these support the membrane from the inside. So that's the purpose. Those two structures help this membrane stay together. Well, now we've got these great big guys. These are all, so everything in gray here, here, here are proteins. And there are tons of proteins in your cell membranes, in your plasma membranes. So I'm going to do a category over here for proteins. So even though each of these is proteins, we have to have a way to talk about them. We can talk about them by where they're found. So we can define these proteins by location. So if we talk about location first, all right, I'm going to pick, I'm going to color code these. Let me see, let me grab light green and so they can be found either inside the bilayer. And so here, I'm just going to color this guy. Those are both found inside the bilayer. Or they can be found on top of the bilayer. So if they're inside the bilayer, they're called an integral protein. They can also be found on top of the bilayer or on the surface of the bilayer. An example would be here. So these guys, they're attached to another protein, but they're found on the surface. So you see here, here. If they're found on the surface, they're called a peripheral protein. And you might have heard these words before. So to integrate, back in the 1950s and 60s, there was segregation in the schools in the South. And the government said you have to integrate, which meant you have to mix, you have to include Black students with the white students. Well, that's what we're doing here. These proteins are included in the bilayer. Peripheral means you're kind of on the outside. So if there's a popular group at school and there's somebody who's kind of in with that group, they're on the periphery of that group. Peripheral means you're at the edge. And so these are peripheral proteins because they would be on the surface. Sometimes, though, these proteins we can also categorize them by shape. So we can talk about their shape. And so we have one example of that, and I'll do this guy in light blue. Oops, hold on, drop my light blue pen. So this guy's got this, basically, it's a channel down through the center or a hole down through the center. So this has got a unique shape. This is called a channel protein, where things that are the right size can just kind of go whoop right through. So it acts kind of like a passage. Not seen here. There are also gated proteins. I'm just going to add this. We don't have them. They're not shown. But they have a passage, but they have something that blocks the top. So again, everything that's here, those are all proteins. And we can talk about them by where they're at or their shape. Sometimes, so for an example, this is an integral channel protein. So we could talk about both its location and its shape. This is an integral, just, non gated protein. The last thing that we have to talk about are these structures. I should have probably saved, this is in blue. So let me just switch to, what color haven't I used yet? I've not used light purple. So we're going to do all of these guys in light purple. Actually, let me move that. I need more space to write. These are called carbohydrate chains. And these are very, very important. If you went to an activity, so when you guys go to Real Money, Real World, or something that you have other schools at, you all wear name tags, and people can tell who you are based on your name tag. Well, how does a cell identify itself? Carbohydrate chains are the name tags of the cell. So they do two functions. They, first of all, if a cell needs something, it will send a carbohydrate chain up, kind of like you raise your hand in class. So the same way a cell would raise its hand, it raise its carbohydrate chain and say, hey, I need something. So the first thing that these carbohydrate chains can do is signal need. So if a cell needs glucose to make ATP, it'll send up some chains, and other things that are flowing by will recognize. So this is kind of the cool thing. In one sense, it's cool, but in another sense, it's a problem. Everything that comes by reads what's going on in the cell through shape. So for example, if I put my car key in my ignition and turn it, my car turns on. If I put my house key in my ignition and turn it, nothing happens. My car can tell that the shape fits. The shape is right. And because that's the right shape, the next thing will happen, which will be the car starting. Well, here's an example. You have white blood cells that roam around outside the cell. And every so often they'll stop and they'll put down, I'm going to do this in pencil because you guys don't need to put this in your notes. I just want to try to kind of make a point. So if I have a white blood cell, it'll put down a kind of a sensor, a protein, and it'll check. So this is my white blood cell here. It'll check the carbohydrate chain, and it knows what carbohydrate chain should be there because your body has a certain set of chains that are allowed, and it'll come through and it'll check the next one, and it'll check the next one. But if it gets to a cell and the cell doesn't have the right carbohydrate chain, this white blood cell will come in and it will kill the cell. So this is where the name tag comes in. These carbohydrate chains provide recognition for what the cell, for the cell's identity. Here's the thing, and this matters. What's going on right now, guys? COVID, and more COVID. Well, the other thing is when the cell signal's needed, it'll put up a chain, and so something else will come along and it'll bind to these chains, and then the cell will bring it in. This is what COVID did. It has a fake protein that looks like it should fit with the cell. And so when it comes along, it clamps its protein down on this carbohydrate chain, and the cell goes, oh yeah, that's what I needed. That's what I wanted. And it pulls not the structure in, but it pulls the virus into the cell. So these carbohydrate chains are huge. It provides the recognition and the need for the cell, but viruses have hijacked it, and they use it to trick the cell into taking it into the membrane. So that's a problem. And scientists are learning more and more about how the coronavirus uses its protein chain to get in. And that's one of the ways they're targeting the treatment for coronavirus. All right, this was our fluid mosaic model. So our takeaways here are we have the phospholipid bilayer as the backbone. We have two structures that help it stay together, cholesterol and microfilaments. These proteins, we can talk about those by where they're at or their shape, integral channel proteins. And then these carbohydrate chains have a huge function. They tell the outside what's needed, and they help identify the cell. It's like its name tag, but it can be tricked. So that's our video. Thanks for watching.